Hey everyone, Dustin Slavine here, and this will be your Module 8 lecture regarding nonfermenters and other gram-negative rods. So here's an overview of some of the topics that we'll be covering today. These include general characteristics of the nonfermenters, ecological reservoirs and epidemiology, common virulence factors, working up of wound cultures, biofilms, and clinically relevant organisms. Now, I want to take some time to reiterate that it is very important that you read your textbook and that these lectures are intended to supplement your textbook readings and other activities. Collectively, this is just meant to give you a better understanding of the how and the why behind things. This will really help you in your career, and also it will help you when you take the BOC, just knowing this background information about things. So here's a general overview of characteristics that are shared by the nonfermenters. All the nonfermenters are obviously gram-negative bacilli or cocobacilli. Most of them are slender, but they vary in size and shape depending on the species. Most are strict aerobes, and all of them do not ferment glucose. Some of them oxidize glucose aerobically, but others are asaccharlytic. Most are catalase positive, and many are moto with polar flagella. And then most of them are also opportunistic pathogens as well. The nonfermenters are ubiquitous in the environment and they can be found in soil, water, plants, and water system biofilms. And the reason for this is for a few different reasons. They have an extremely versatile metabolism in that they do not rely on carbohydrate fermentation. Instead, they use aerobic respiration and they can metabolize a wide variety of organic compounds. More specifically, they can use trace organic molecules found in water, plastics, detergents, and disinfectants, and many are able to degrade surfactants, hydrocarbons, and preservatives. Because of this metabolic flexibility, they're able to survive in nutrient-poor environments where many other bacteria may not. Additionally, they are oligotrophs, meaning that they can thrive in very low nutrient environments. Examples of this would be distilled or tap water, sink drains, humidifiers, hospital water systems. They can even persist on very small amounts of carbon from rubber tubing, plastic containers, and trace contaminants in water. Another important thing to note is that many of the nonfermenters show natural resistance to antiseptics and disinfectants due to outer membrane permeability barriers, efflux pumps that pump out the toxic chemicals, enzymes that are able to degrade antimicrobials, and also biofilm protection. This allows survival in solutions that would kill many other bacteria, including diluted disinfectants. So for these reasons, they are strongly associated with nosocomial infections. It should be noted that these organisms often colonize patients without causing overt disease. Here I've listed some of the common virulence factors shared by the nonfermenters. They include adhesion factors, capsule and slime layer, exotoxins and toxins, proteolytic enzymes, pigment production, endotoxins, siderophores, and antimicrobial resistance mechanisms. So, adhesion factors are able to promote colonization because they mediate initial attachment to host tissues. A capsule is made up of polysaccharides which surround the bacterial cell, and these allow bacteria to resist phagocytosis. And also, they help mediate drying-related cell death, essentially. Exotoxins and toxins interfere with host cell processes such as protein synthesis and signaling pathways. Proteolytic enzymes enable bacterial spread in tissues by degrading extracellular matrix proteins and host defense proteins. Some bacteria, such as Pseudomonas aeruginosa, produce pyoveridin and pyocyanin, which contribute to oxidative stress and iron scavenging. Lipopolysaccharide triggers host inflammatory response, fever, and septic shock in severe infections. As mentioned in the previous module, siderophores compete with host proteins such as lactoferrin and transferrin to sequester iron from the host. And then non-fermenters tend to have a lot more antibiotic resistance mechanisms, which prolong infection and allow biofilms to persist. I thought it would be good to take some time to talk about the workup of gram-negative rods in wound cultures, due to the fact that non-fermenters are important wound pathogens in that they are abundant in moist environments, they survive well in nosocomial settings, they're capable of forming biofilms, they produce tissue-damaging virulence factors, they exhibit high antibiotic resistance, and they also act as opportunistic pathogens in damaged tissues. Although clinically relevant gram-negative rods in wound cultures are not limited to the non-fermenters, I think it's important to talk about this because coming from someone who has worked in a few different types of laboratory settings, I've found that many smaller hospitals that perform microbiology, they tend to struggle with working up mixed cultures and determining what's clinically significant versus not. So the first thing to talk about in relation to this is optimal specimen type. Optimal specimen types would include biopsies and specimens that are aspirated by needle because they provide higher diagnostic yield due to the lack of superficial flora. They improve the preservation of delicate organisms, and they are able to access deep infection sites. And as such, they reduce the rate of false negatives. Swabs are indeed a viable option and probably the easiest to collect for providers, but they're frequently suboptimal, especially depending on the culture type. They only sample the surface, which results in a much lower sample volume, and there is also a risk of drying to the specimen, which can affect overall organism recovery. To be more specific, they are poor samples for anaerobic culture and mycobacterial culture especially. And due to the presence of superficial flora, they can kind of muddy the waters per se, and they are kind of like a source of quote unquote noise for the culture. And by that, I mean that they can contain mixed microbiota, and you're not able to tell whether they're, transient residential flora or if it's truly a pathogen, especially when the infection's polymicrobial. This is why performance of the gram stain is critical, because it can help guide you. The presence of white blood cells and epithelial cells in the gram stain smear help assess specimen quality. Many leukocytes indicates inflammation, and the lack thereof may suggest contamination or absence of cellular material, especially when epithelial cells are also absent. However, depending on the specimen source, sometimes epithelial cells are not expected to be present. So something that I've mentioned in past lectures is that you want to focus on the predominant organism types or predominant organism. And if there's multiple that are predominant, especially when you have multiple gram-negative rods that are mixed and predominant, then maybe you'll want to consider that this is either a polymicrobial infection or, the sample is contaminated with mixed microflora. So interpretation of these cultures is heavily reliant on clinical correlation. So providers will correlate the culture report with the gram stain results, and if it's present in the culture but not present in the gram stain, then that sometimes reflects low-level colonization or contamination. And then you'll want to look at the relative abundance of organisms in the culture, like I mentioned, and the likely pathogen should match the gram stain, host response, and clinical symptomatology of the patient. Another thing that they'll look at is historical epidemiology. Does the organism correlate with the type of infection observed or clinical presentation? Does the patient have a history of this type of infection? They'll also they'll take multiple cultures from one collection site, and they'll consider other lab results too. Because if it's present in multiple culture sites, then perhaps this is a problem. Or if it's present in maybe one and not the others, then maybe they'll think that it's low-level colonization, or perhaps it's a contaminant. Local signs of infection include erythema, edema, pain, warmth, purulence, and delayed healing. And systemic signs of infection include increased fever, an elevated white count, an elevated CRP or ESR, as well as other lab results, such as lactic acid. They'll also look at the other risk factors for the patient, such as their immunologic status and medical conditions that might lead to defective defenses. And then they'll also look at how the patient responds to treatment. So now I want to switch gears and talk a bit about biofilm formation. So a biofilm is a community of microorganisms that's attached to a surface and embedded in a self-produced extracellular polymeric substance matrix. EPS consists of polysaccharides, proteins, lipids, and extracellular DNA. And these cells within this biofilm communicate via quorum sensing. Quorum sensing is a cell-to-cell communication mechanism in bacteria in which they produce and detect signaling molecules to coordinate gene expression when population density reaches a certain level. It's important because quorum sensing helps regulate bacterial virulence, biofilm formation, and antibiotic resistance, which helps explain how the pathogens become more pathogenic during infection and why some infections are more difficult to treat. So these microorganisms may adhere to biotic surfaces such as teeth or abiotic surfaces such as catheters, implants, surfaces, etc. And they are oftentimes responsible for persistent or device-associated infections. The stages include initial attachment, irreversible attachment, maturation, and dispersion and detachment. It is important to note that device-associated infections may initially culture negative because organisms are embedded in the biofilm rather than freely planktonic within the specimen. So the purpose of this slide is to provide a little bit more context into biofilms. As I mentioned in the last slide, biofilm-embedded sessile cells may not disperse well during specimen processing, and that results in false negative cultures or reduced colony counts. Standard MIC testing measures the susceptibility of planktonic cells, which often underestimates resistance of organisms and biofilm-associated infections. In fact, in mature biofilms, they are 100 to 1,000 times more tolerant to antimicrobials due to limited drug penetration, metabolic dormancy, and the protective matrix. The high cell density present in biofilms is favorable for horizontal gene transfer, which facilitates the rapid exchange of resistance plasmids such as VAN-A and MEK-A. Quorum sensing regulates biofilm maturation, virulence expression, and toxin production once a critical population density is achieved, and in nature, greater than 90% of organisms exist within a biofilm. So now let's take some time to talk about the clinically relevant nonfermenters. Pseudomonas aeruginosa is beta hemolytic and oxidase positive. In fact, it is the number one most common non-fermenting gram-negative rod isolated in the clinical laboratory. It is capable of growing at 42 degrees centigrade, and it may produce a metallic sheen. Additionally, Pseudomonas aeruginosa is said to have a grape-like odor or a corn tortilla odor, depending on the strain. It may possess a blue-green pigment, pyocyanin, and a yellow-green pigment, pyoverdin, and the production of pyocyanin within a strain is diagnostic for Pseudomonas aeruginosa. Mucoid colony types of Pseudomonas aeruginosa are hyperproducers of alginate, and this is especially implicated in cystic fibrosis patients where these strains are more likely to be found. Pseudomonas aeruginosa, kind of like Staph aureus, is considered to be a universal pathogen, and its presence is generally always reported in aerobic cultures unless it is heavily mixed or found in a stool culture. So here I made a table of some of the most commonly found Pseudomonas species in the clinical laboratory. These include Pseudomonas aeruginosa, of course, but also Pseudomonas fluorescens, Pseudomonas putida, Pseudomonas stutzeri, Pseudomonas mendocina, Pseudomonas luteola, and Pseudomonas oryzahabitans. For the boards, I would say you probably want to know about the first four, Pseudomonas aeruginosa, fluorescens, putida, and stutzeri. Pseudomonas aeruginosa has pyocyanin and pyoverdin. It can grow at 42 degrees centigrade, and it has a sweet grape-like odor or corn tortilla odor, and it is beta hemolytic on blood agar. Pseudomonas fluorescens generally has a yellow-green color due to the presence of pyoverdin. It is commonly an environmental contaminant, and it grows better at 25 to 30 degrees centigrade. Pseudomonas putida usually has low virulence, and its colony type is generally like a yellow color. And then Pseudomonas stutzeri tends to be dry, and it's non-pigmented or light brown. So now let's talk about Stenotrophomonas multifila. This organism is oxidase negative and motile with polar flagella. It is the third most common non-fermenting gram-negative rod isolated in the clinical laboratory. It is intrinsically resistant to many beta-lactams and also to the aminoglycosides. Trimethylprim sulfamethoxazole, levofloxacin, and minocycline are the drugs of choice for this organism. And Stenotrophomonas multifila is implicated often in cystic fibrosis patients with chronic airway infection, as well as patients with neutropenia and or a compromised immune status. It should be noted that among the Stenotrophomonas species, Stenotrophomonas multifila is the only one known to cause human disease. So the most important organisms from the Acinetobacter family are from the Acinetobacter baumannii calcoaceticus complex, or the ABC complex. This includes Acinetobacter baumannii, Acinetobacter calcoaceticus, Acinetobacter pittii, and Acinetobacter nosocomialis. These organisms are all phenotypically similar, so they are bunched together in this complex. Acinetobacter baumannii is almost exclusively associated with hospitals, and it's generally associated with severe trauma and with immunocompromised individuals. It is capable of causing respiratory disease, urinary tract infections, meningitis, and during the Iraq War, many soldiers were afflicted by this organism, and it was given the nickname Iraqibacter. Members of the ABC complex are particularly problematic because they are beta-lactam resistant due to the presence of Bla-imp or Bla-oxa. As such, this group is generally treated with tetracyclines or glycocyclines or polymyxins. However, 1 to 2% of isolates are colistin resistant due to the complete loss of lipopolysaccharide. Other species that are not part of the ABC complex include Acinetobacter lowolfii, Acinetobacter bailey, Acinetobacter lacticae, and Acinetobacter radioresistens. These organisms are less virulent than the ABC complex, and they're often considered normal flora or transient flora, and you do not need to know these for the BOC. However, you will encounter them if you work in a clinical microbiology laboratory. All of the Acinetobacter species are cocobacilli. Sometimes they're gram variable, but usually gram negative, and they are non-motile, oxidase negative, and catalase positive. Achromobacter species are oxidase positive and motile with peritrichous flagella, and the most important clinically relevant species is Achromobacter xylose oxidans. Other members of the Achromobacter genus include Achromobacter denitrificans, Achromobacter spanius, Achromobacter pieciaudii, and Achromobacter marpiatensis. And these are difficult to resolve to the species level by MALDI-TOF, so they are often reported as Achromobacter species group. Achromobacter species are commonly resistant to aminoglycosides and many cephalosporins, and they are often misidentified as Pseudomonas species or Burkholderia species. However, this happens a lot less now because of MALDI-TOF, but in laboratories that are using biochemicals still as their main form of identification, this misidentification does occur. And Achromobacter species are important pathogens in cystic fibrosis patients and immunocompromised patients, as are many of the other non-fermenters. So I didn't put these two together on this slide for any particular reason. There's just less to know about them for the BOC. First one I'll talk about is Alkalogenes faecalis. It is oxidase positive, catalase positive, and negative for nitrate reduction. And one of the most commonly noted characteristics of this organism is that it has a fruity odor. Most say that it smells like green apples. The colonies are non-pigmented, flat, and spready, but they are not swarming. This is typically a low virulence opportunistic pathogen with variosusceptibility, but it may show resistance to many beta-lactams and aminoglycosides. Bordetella bronchoseptica is positive for oxidase, catalase, nitrate reduction, and it's rapidly positive for urease. And unlike other members of the Bordetella genus, Bordetella bronchoseptica will grow on conventional media such as blood agar. Bordetella pertussis and parapertussis do not grow on conventional media. Usually this requires Bordet Gengao or Regan low agar. Bordetella pertussis is differentiated from bronchoseptica because pertussis is non-modal and urease negative. And it is important to note that Bordetella holmesii may also cause whooping cough like other members of Bordetella. The colonies of Bordetella bronchoseptica are small, smooth, and grayish, and dogs, cats, rabbits, and pigs are the primary reservoir. Infections are generally found in patients that are immunocompromised or in patients that have known animal exposure. Next, we're going to talk about Chromobacterium violaceium, which is one of my favorite organisms. It is violet due to its characteristic pigment violacein. Chromobacterium violaceium is also a facultative anaerobe, unlike other members of the non-fermenter family. It is oxidase positive, catalase positive, and modal, and this organism is generally found in the environment in soil and fresh water in tropical or subtropical regions. And patients are usually exposed to contaminated water or soil, and often through traumatic wounds. Generally, this is limited to a wound culture pathogen. I've only ever really seen it in wound cultures. I haven't seen it in urine or anything, but I'm sure it's happened before. This organism is rare but highly virulent, and mortality can be due to metastatic abscess formation. Chromobacterium violaceium is multi-drug resistant, but it is generally susceptible to the fluoroquinolones such as levofloxacin and ciprofloxacin, as well as carbapenems and trimethylprim sulfamethoxazole. Because you need to know these organisms probably a lot less for the BOC, I've kind of grouped them all together under Flavobacteriaceae. So the Flavobacteriaceae are oxidase positive and catalase positive, and many of them produce a yellow to orange pigment, which is due to flexirubin type pigments. They are often environmental contaminants but can cause serious opportunistic infections, and they are known for their unusual antimicrobial resistance patterns, meaning they're multi-drug resistant and sometimes hard to treat. Important genera within the Flavobacteriaceae include Elizabeth kingia, Myroides, Weichsella, Burghiella, and Chryssobacterium. Myroides typically has a fruity odor and a yellow pigment. Elizabeth kingia is indole positive, but it is rarely isolated, has a high mortality rate, and there's only 200 to 500 cases in the US annually. Reduction of mortality is associated with proper identification between Elizabeth kingia meningoseptica and Elizabeth kingia anopheles. Weichsella is often isolated from genitourinary specimen types. Burghiella zoohelcom is associated with animal bite infections, and Chryssobacterium is identified by order of exclusion, meaning it doesn't really have any specific characteristic that makes it stand out from all these others. It just means that you ruled everything else out, so it's probably Chrysio. And MALDI-TOF has improved the means of identification immensely and reduced mortality risk due to misidentification by biochemical systems. The Burkholderia cepacia complex includes Burkholderia cepacia, Burkholderia senocepacia, Burkholderia multivorans, Burkholderia vietnamiensis, Burkholderia dolosa, and Burkholderia gladioli. If Burkholderia gladioli is isolated in a cystic fibrosis patient's culture, then it should be sent for a confirmatory ID. All of these organisms, when found, are reported as a Burkholderia cepacia complex. These organisms are oxidase positive, catalase positive, and motile with polar flagella. Pseudomonas cepacia selective agar is used for the isolation of Burkholderia cepacia complex in cystic fibrosis cultures. This organism is a important pathogen in patients with chronic granulomatous disease or cystic fibrosis. This complex is known to be intrinsically drug resistant to many drugs, and a recent development regarding this organism is that there are no longer any CLSI guidelines due to poor reproducibility and correlation of the MIC with actual patient response. Testing now focuses on identifying resistance mechanisms instead, such as R&D efflux pumps. The Burkholderia cepacia complex is associated with nosocomial infections. So now let's talk a bit about Burkholderia pseudomallei, which is a BSL-3 bioterrorism agent. This organism is the causative agent of melioidosis, and it may present as a severe pneumonia, septicemia, and it may result in abscess formation or a Mycobacterium tuberculosis-like syndrome. This organism is endemic to Southeast Asia and Northern Australia, and it is linked to exposure to soil-contaminated water or occupational exposure to rice paddies and farming. Routes of transmission include percutaneous inoculation, inhalation, or the ingestion of contaminated water, and predisposing conditions include diabetes, alcoholism, and chronic kidney disease. When suspected isolates are found, they must be immediately evaluated by performing rule-outs. They should not be definitively identified. And if your lab does not have the capability of performing rule-outs, then it needs to be immediately referred to a reference laboratory. And not like a regular reference laboratory, I'm talking about like a public health laboratory. One of the important factors or characteristics of Burkholderia pseudomallei is that it exhibits polar safety pin staining upon gram stain. So here I've listed some characteristics of Burkholderia pseudomallei and the rule-out cascade that you would perform if you were suspecting it. Another important bioterror agent of the Burkholderia genus is Burkholderia mallei, which is the causative agent of glanders. This is generally a zoonotic infection, primarily affecting horses, but also mules and donkeys. Generally, patients have direct contact with infected animals or tissues, and this is more commonly found in Asia, the Middle East, or South America. It's pretty rare in the United States. Human disease presents as a cutaneous, pulmonary, septicemic, or a chronic formation of abscesses. And similarly to pseudomallei, when you suspect it, you must immediately try to rule it out. And if you can't, then you need to refer it. And this organism also exhibits safety pin staining upon gram stain. And then here I've shown you the rule-out cascade for Burkholderia mallei. Switching gears a little bit, we're going to talk about some of the more unusual non-fermenting gram-negative rods. So first, let's talk about Vibrio species. Vibrio are gram-negative curved rods that are said to be comma-shaped. They are facultatively anaerobic, oxidase positive. They exhibit rapid darting motility, and they're all halophiles. They are found in marine and brackish water, and generally infection occurs when a patient is exposed to undercooked seafood or they have exposed wounds in salt water. The media type used for isolation and identification is thiosulfate citrate bile salts, or TCBS agar. One of the most notable species is Vibrio cholerae, which is the causative agent of cholera, and this is characterized by the presence of rice water stools. And rice water stools, they are pretty much how they sound. It looks like rice water, and this is due to the hypersecretion of water and electrolytes. And some patients report having 15 to 30 bowel movements per day, and the risk of mortality is due to dehydration. There's a significant rise in isolating of Vibrio species, and this is due to the increase in ocean water temperature, travel to coastal or cholera endemic areas, increased consumption of seafood, increased use of recreational water facilities, larger populations of immunocompromised individuals. Helicobacter pylori is a curved helical or spiral or fusiform gram-negative rod that is said to be S-shaped. It is a microaerophile and a rapid producer of urease, and it is motile with a rapid corkscrew motility. Helicobacter pylori is known to colonize the stomach and cause low-grade inflammation for a long duration. But it should be noted that approximately 50% of the world's population is colonized with Helicobacter pylori, and this does not cause overt disease. However, of this 50%, 10 to 15% develop gastro or duodenal ulcers. Sometimes this is known as peptic ulcer disease. And in 1% of patients, gastric carcinoma or mucosal associated lymphoid tissue lymphoma occurs. Helicobacter pylori is thankfully highly responsive to antibiotic therapy, and after antibiotic therapy, I think it's usually about 3 weeks or so of treatment, then you retest the patient, and then hopefully their test is negative at that point. Diagnosis is generally made by breath test primarily. However, it can also be made through histology stains of gastric biopsies and molecular methods. The CLO test was formerly used but discontinued due to poor sensitivity and the risk of false negatives in patients undergoing proton pump inhibitor therapy, such as drugs like omeprazole, which many patients do take for, gastroesophageal reflux disease. This diagram depicts how the breath test for Helicobacter pylori works. The urea breath test detects infection based on the organism's strong urease activity. And basically what happens is the patient ingests urea labeled with carbon 13 or carbon 14, and if H. pylori is present in the stomach, the urease enzyme breaks down the urea into ammonia and labeled carbon dioxide. The labeled carbon dioxide is absorbed into the bloodstream and exhaled in the breath, where it's collected in a mylar balloon and measured. Campylobacter species are gram-negative curved or S-shaped rods that are sometimes spiral, and many say that they resemble a gull wing, like they look like seagulls on gram stain. Campylobacter species are microaerophilic, meaning they require little oxygen to grow. And it is important to note that they generally require a much higher temperature for incubation, usually around 42 degrees centigrade, which means that they are a thermophile. Campylobacter exhibits darting or corkscrew motility, and they are oxidase and catalase positive. In the United States, Campylobacter jejuni is the most frequently isolated in clinical specimens, and post-infection, this organism is associated with Guillain-Barré syndrome, which is an autoimmune disorder where the immune system damages peripheral nerves and causes rapid ascending muscle weakness, tingling, and even potential paralysis. Other clinically relevant species include Campylobacter foetus, Campylobacter coli, and Campylobacter ureolyticus. The Campylobacter species are commonly found in poultry, cattle, and contaminated water, and they are more commonly found in the summer, meaning infections are more prevalent in the summer. Generally, transmission occurs by the fecal-oral route, and it should be noted that Campylobacter is the fourth most common cause of foodborne illness in the United States, and it is the most common cause of bacterial gastroenteritis. However, this organism does not multiply in food. Thank you for taking the time to watch this lecture. I hope you learned something today. Take care.